Like everyone else, I'm very happy to be here. The welcome to Washington yesterday was a slight case of food poisoning, so I'm really happy to be here. I represent the Palmer LTR, and I wanted to start with this slide because it tells a lot about the system in which I've been working in since 1974. When I first began working here as a graduate student, the glacier you see in the background had a grounding line. It covered this penguin colony. And in the last 50 years, that glacier has retreated about 500 yards. That penguin colony, which was positioned originally in front of the glacier, was completely covered in Adelie penguins. The paleoecological record indicated that colony had been there for at least 1,000 years. The presence of Gentoo penguins in that record, well, there is no record of Gentoos. They moved in 1993, and ever since that time, their populations have taken off. Adelie penguins have decreased in our region by 85%. Gentoo penguins are up 33,000%. So what I want to talk to you about, I hope when you leave here today, you're going to have a better appreciation for why these changes have occurred. By way of background, where we're located, we're in a very big and empty ocean. The inception of our program was in 1990. Although, like I said, our work began in 1974. We have 10 investigators, 8 institutions, and we sample annually a marine domain of about 150,000 square kilometers in 20-kilometer grids. That grid is anchored by Charcot Island on the south, one of the most godforsaken places you could ever find, and on the north by Palmer Station, and west to the continental shelf. That makes up roughly the spatial extent of the grid. Palmer Station is one of 3 stations that are under the supervision of NSF. Palmer is the smallest. As you can see, it looks like it sits out in the middle of nowhere, and it actually does. It's perched on a rock called Gamage Point. The term point might be a misnomer because glacier deformation in the back is indicating that Palmer may actually be on an island. So time is going to tell on that one. It is, like I said, the smallest station. Our entire capacity is 43 people, 45 if you stretch it. We deal with a challenging environment. We do dangerous work in a dangerous place, but there's also social challenge to working at Palmer. 43 people, 24/7, for months on end. Let your imagination take over there. In terms of how we sample our system, Palmer, ironically, even though it's very close to South America, it's only 800 miles away, is only accessible by ship. So it is logistically the most isolated of the Antarctic stations, and as a result, fairly remote. The ship you see in the background is a Lawrence M. Gould, which serves to do everything. Carry cargo, fuel, food. It also serves as a research vessel that samples that grid. So what I'm going to talk about today, and I hate to admit this, is failure. And it's the failure of one life history strategy, naming that of Adelie penguins, where they're a polar species, they depend on sea ice, compared to what is happening to Gentoo populations. I've already mentioned the tremendous decrease in Adelies compared to the tremendous increase in Gentoos. And one of the main objectives of the Palmer LTR, what unifies us, is our interest in understanding how sea ice, the presence and absence of sea ice, affects the Western Antarctic marine ecosystem. And we also have the same interest that we sample the ocean through the life histories of these birds. So much of our work has really encompassed learning everything you would ever want to know about penguins and their life histories. And hence the title of the slide. But when life histories begin to fail, that usually signifies a change in the environment. And those are the linkages that we're looking for. I'm going to start with a short video. As you can see, our system where we work is really different than our colleagues in the French Polynesia. But I'm going to start with a short video of us collecting life history data early in the season, just as breeding gets underway. And you're going to notice a human being with big goggles on that does not move during the video. She's actually alive, but 8 days prior to this video being taken, she was in her native Tanzania in 120-degree weather, and she was working in 10-degree weather. So anyhow, let's see if this will roll. This is what our technicians do when they're shut in. Beautiful videos with incredible music. But anyhow, so I don't lose time, I want you to pay attention to the landscape in which we're working.
Washed my face in the rivers of Empire.
And the colony above this
Made my bed from the cardboard crate.
completely surrounded by snow.
Down in the city of
What
Court.
we're doing right now is our first census.
No
our
news,
first
no
idea of
new
the first
regrets.
and
Tossed a Susan B over my shoulder. Prayed it would rain and rain. Submerged the whole western states. Call it a last fair deal
You'll see we put a green towel
with
down
American steel and corporate handshake.
A very interesting observation is that recent analyses have shown that the egg weight of the Adelie penguin female is linked to the sea concentration effect on sea ice. Really exciting analysis. Scaling up to a global perspective.
Take the story of Carpenter Light. He dropped his tools and his keys and left, and headed out as far as he could.
Also want you to pay attention to these
Past the city and gated neighborhoods. And slept beneath the stars. Wrote down what he dreamt. And he built a machine for no one to see. Then took flight, first light of new morning.
Shove the egg back underneath them. And then after we're done with one nest, the pairs reunite, everything's fine. So, what's the big deal with this? We've been collecting these data for a very long time. And one of the things that we discovered is that even though Gentoo penguins are subantarctic, it still maintained their breeding chronology in what was a polar system. And what that does is a shift in chronology between Adelies and Gentoos. Gentoos do everything that Adelies do, but they do it 2 to 4 weeks later in terms of the timing of their breeding. And why should this be important? Well, it turns out that in Antarctica, because of warming, when you remove sea ice off the ocean, it vents moisture into the atmosphere. So snow has been increasing on the western Antarctic Peninsula for at least 100 years. A paper that just came out 2 weeks ago said 200 years. Whatever it is, there's a lot more snow falling there over the last few decades. The snow falls on existing snowpack near these Adelie penguin colonies, especially in the spring. We get one blizzard after another. These birds, in some cases, you can't even see these birds. They're completely covered by snow. Unfortunately, these big blizzards are also accompanied by major shifts in the wind that come from the north and begin to instantly warm this fresh, wet snow, and this snow melts. And this is the result. This is the colony that we were working in 2 weeks previous that we were looking at. This is what happens during these melt events. It's 100% mortality of the eggs, usually. And I know that anyone that's ever wanted to know how smart a penguin is, there's a bird in the background sitting on a stone nest trying to keep an egg warm in 6 inches of 32-degree water. So they are hardwired into beyond description. They just will not abandon their eggs. Getting back to this breeding chronology thing, there's no question that these enormous snow events are now affecting the populations of Adelies, driving them down. And we've developed landscape models that indicate that on some landscapes, snow events alone are actually driving populations to extinction. The 2 pictures up on top, another effect that we've noted is that eggs that do survive and produce a chick that essentially grows in an environment that is this moist can't thermoregulate. And their growth rates slow down to the point where when they're ready to leave their parents and head for the ocean, they're not heavy enough to survive winter. So it's a pretty all-encompassing effect, termed mechanistically. I'll switch gears now a little bit and get into our second failure, life history failure. And one thing that we learned from this, we discovered this landscape effect, as we're calling it, pretty early on. One of the things we learned from that experience is that in terms of looking at penguins, we were looking at things on very large scales, when in fact it's these subtle differences in life history that seem to determine the winners and the losers in a warming Antarctic peninsula. And looking at this figure here in pretty simple terms, this is a large-scale picture of sort of what we thought about the system around the year 2000. So, you've got temperatures increasing, you've got sea ice decreasing, and you've got Adelie penguins decreasing and Gentoos increasing. We knew by then that Adelie penguins were ice dependent, Gentoos were ice intolerant. We had enormous databases in which we could document, chick weights, all these variables that we could see were linked to sea ice or open water. That we didn't know what the translation was. I think Dave this morning talked about mechanisms or somebody, that we just did not know how exactly is a presence or absence of sea ice translate into a demographic signature. Pretty frustrating position to be in. Why NSF kept funding us, I don't know. But we started thinking, if there's really subtle life history differences or making a difference. Maybe we're just looking at this in the wrong scale. What we need to do is scale down and understand the ocean like our penguins perceive it. And that's what we started doing. Some of the issues that we had prior to the year 2000, you can guess at some of these already. We have this emphasis on terrestrial studies, that penguins spend most of their lives at sea. We didn't have technologies to deploy that could like satellite tags or something they could put on a penguin. We also realized through a lot of this long-term data that penguins operate over really small scales compared to oceanographic studies. And one of the reasons I should mention is, you see the circle there around a flipper, penguins can't search the ocean like an albatross. They got little flippers, they just can't move very far. And so we started to get this idea that we needed to just scale down what we knew about how these birds foraged at sea, in the hope of finding the link to sea ice. So how did we do this? First of all, this Anvers Island, on the lower panel there, is where Palmer Station is located. Around Anvers, we have the two species of penguins of interest. We have enormous numbers of colonies, but that's the distribution of the populations. We knew from some very primitive instruments we had, little tiny VHF transmitters, that the foraging range of these birds was probably not that large. And in fact, it's proven to be the case. They only forage about 15 to 20 kilometers away from their nest site. Now, why they forage in Palmer Deep Canyon goes back to this idea of they can't search the ocean. The way penguins have solved the problem of not being able to search the ocean is by breeding next to these areas of very high predictable production. And these deep canyons that cross the shelf break are those sites. There's a constant input of nutrients that comes in from the continental shelf and upwells almost exactly in front of these penguin colonies. So there's this immense production, lots of food over predictable periods of time. And by that I mean ecological time, decades to centuries, which is roughly what it takes to keep a population going. So we realized we were in a perfect experimental setting. How are we going to approach it? We were really lucky that when we began to realize this, colleagues that we were working with had managed to miniaturize satellite tags. Up until that point, the satellite technology was there, but it was great on a polar bear. It wasn't really that good on a bird that only weighs 10 pounds. So this became one of our main instruments to begin this exploration process. This was revolutionary for us. Next, we decided that instead of looking at sea ice as some sort of impediment to what we were doing, we needed to take more advantage of this situation. And we began to record and log data at the scales, the sea ice data at the scales that the penguins were using the ocean. And let me show you how variable sea ice can be at Palmer Station. This is a 15-second video of a 2-hour event. This happens constantly. This ice that moves in and out in different densities and different kinds. But it's super variable. So we began thinking, okay, we're going to start documenting our information in relation to what's happening to sea ice. The sea ice, fortunately, can persist for sometimes days, weeks, months. It really, really aligns very well with the foraging ecology of these birds in terms of this particular variability I'm talking about. And finally, in 2008, we got a new co-PI in our program, Oscar Schofield, who leads one of the most sophisticated labs in the United States out of Rutgers University insofar as using robots to monitor the ocean. And you notice the acronym RU-Cool. If you knew Oscar, you would know why that acronym is there. And also my collaborator at Rutgers, Josh Kohut. So we work with both of them. Now what did we do? Well, first of all, we had enormous amounts of satellite data. And these data can be downloaded every 24 hours and sent to the RU-Cool lab. The RU-Cool lab can then pick up a phone and program the gliders that are working over the Palmer Deep. And so within 24 hours after a penguin goes out on a foraging trip, we can program a glider to pretend it's a penguin and cover the ocean exactly like that penguin did. Getting fine-scale information on what a penguin sees doesn't get any better than that. What have we discovered? By scaling down. Pretty fascinating. The long story shortened into 2 slides. First of all, like I said earlier, when sea ice no longer covers the ocean and it's moisture, when sea ice no longer covers the ocean, it's also subject to wind mixing. Without sea ice, the wind really whips the ocean up. It's called the mixed layer depth, the upper boundary of that turbulence. When mixed layer depth, and by the way, the large panel there, you can see this a couple relationships I want you to see. One of them is that the ocean has become much more variable since about 1994 in terms of where the amount of sea ice and the mixed layer depth. Huge variability. The other is that when there's more sea ice, the mixed layer depth is shallow. And the reverse happens when there's less sea ice. What we also discovered is that the entire food web that tracks mixed layer depth. So phytoplankton, krill, and fish, at least krill and fish, the prey on which these penguins depend, track this food web. In the absence of ice, deep mixing, the food web is much deeper than it is otherwise. This was, to us, an important enough discovery. And by the way, I'm talking about something that is, we just made these connections in the last 3 months through analysis. We're actually holding a workshop in Montana to figure out some papers. So this is brand new info. Now, why is this important? Why is a deep food web important or a shallow food web important? Well, it turns out that Adelie penguins don't dive below the mixed layer. We don't know exactly why. We have some ideas. Whereas Gentoo penguins don't care where they feed. They can be up or down. They dive to 200 meters and it doesn't seem to bother them. So here we have sort of the first evidence, kind of like the first synthetic evidence that without ice, Adelie penguins are essentially being forced to forage inefficiently. And that is cascading across their life histories. Chick weights, in fact, we've already done these analyses. Chick weights seem to be determined by mixed layer depth. Foraging trip durations, all these variables that we've been holding kind of in a box like pieces of a puzzle now have a synthesis point. And I suspect this idea will be the subject of our next proposal, which is due in 2020. A summary of the summary. You can think of Adelie penguins as hardware, Gentoos as software. Adelie penguins are no longer capable. Their life history simply can't handle a wetter, windier, snowier nesting environment in a much more turbulent ocean. A final thought, going back to the initial photo in this, at the beginning of the slide, is Gentoos have taken over the world down there in less than 25 years. That's unbelievable change. If they're tracking environmental change, which they are, 25 years is a really, really short period of time. And I would suggest we keep that number in mind when we think about what's really happening globally in terms of environmental change. What could change and how quickly? I'm not sure we all can capture that. Acknowledgements. There's 50 years of support here almost. I don't know, I've probably forgotten some of the agencies. NSF has been paramount in supporting us. NOAA, NASA, USGS, Department of Defense, lots of private donors, several programs going back to my graduate years, lots of contractors. Most of all, I want to acknowledge the people that have worked for me. This has never been a one-man show. And I've had the pleasure to work with some of the most overworked, underpaid, and happiest people I've ever met. So, thanks very much.
Hi, Lauren Fields, NOAA Fisheries. Regarding the penguin foraging slide that you showed, do the diets of Adelies and Gentoos differ significantly, or are they both feeding on krill and silverfish?
They're exactly the same. They are exactly the same. That's a good observation. It's not being driven by diets. I mean, that's the interesting point in the story is you've got these very similar species that have these very subtle differences in what they can do, and that seems to cascade across their populations.
We have time for some more questions.
So can you suggest, you had some landscape modeling, are there areas that sea ice is increasing and that the Adelie penguins can potentially move to? I mean, do you see any of that across the region?
Yeah, we have no evidence that the birds are moving. They're simply dying out regionally. There was a recent paper, they discovered 750,000 breeding pairs of Adelies on the Weddell Sea side, so the eastern side of the peninsula. The paper was kind of sensationalized a bit. There's lots of reasons why you would suspect there'd be these huge colonies there, because the Weddell Sea gyre is constantly buffering that area with new sea ice. With respect to your do you want to know about this model? I mean, I think I've got a slide I could show you what the model is.
I was just wondering if it has some of that potential movement of the species around there, if with the Gentoos coming in or the Adelies going around.
No, everything is happening sort of in situ. It's local dynamics.